Thank you. We sincerely appreciate your interest in hearing from us about critical considerations when choosing an RT-PCR assay for detection of the SARS-CoV-2 virus. PerkinElmer was one of the first organizations to develop and deploy nucleic acid detection kits in response to the disease outbreak. We would like to take this opportunity to share our experience in assisting people on the front line of fighting the virus. PerkinElmer offers nucleic acid and immunoassay-based solutions for SARS-CoV-2. This session will be on nucleic acid-based testing. We will start by discussing the critical success factors for implementing SARS-CoV-2 testing solutions in labs, followed by introduction to the different components of PerkinElmer's nucleic acid detection workflow as it relates to viral RNA isolation, PCR setup, and finally, the assay for nucleic acid detection itself. Viral dynamics in infected patients are yet to be fully understood. A publication in The Lancet back in March reported that viral load tends to correlate with the severity of infection. Other reports support the possibility of SARS-CoV-2 transmission from persons who are pre-symptomatic or asymptomatic. Therefore, a highly sensitive RT-PCR assay with the ability to pick up low viral load reliably is critical to accurately diagnose and reduce the spread of infection. In response to a survey conducted by the Association for Molecular Pathology, labs report that supply chain interruptions have had a significant impact on their testing capacity, with over 85% of labs reporting that these interruptions have had negative effects, such as delay in turning around test reports and ultimate decline in test requests received over time due to their inability to meet turnaround time obligations. While all lab types were reporting challenges, academic medical centers and community hospital labs reported the most severe barriers, including limited supply of testing kits and reagents. Such an overwhelming message from over 200 responses across all lab types indicate that a steady supply chain is needed to implement and sustain routine testing. The same survey also reported that sample throughput ranges from a few hundred tests to greater than 500 tests a day. Over 90% of the labs plan to increase throughput in the next 2 to 3 months by adding platforms, shifts, or expanding workforce. With the fall season fast approaching in the US, there is an increasing need to run a flexible operation to stay in sync with fluctuating demand. Automation is a critical need to accomplish this without increasing staff and their exposure to the risk of lab-acquired infections during high-volume processing. Lastly, it is very important to find the right partners to ensure that all components of the operation work in harmony with each other. Very often, providers with a broad portfolio covering multiple components of the workflow simplify life for the lab manager. They offer a simplified ordering process, better visibility to availability of materials required for sample processing, and dependable technical support through a single point of contact. At a high level, there are 3 steps in the nucleic acid testing process: sample collection, sample isolation, and nucleic acid detection. The Chemagic 360 platform is PerkinElmer's magnetic bead-based isolation method that is ideally suited for automated sample processing, which our customers have communicated as being extremely important in such emergencies. Sample isolation plate setup and PCR setup can be facilitated by modular JANUS workstations. Automated processing culminates in the detection of the virus using the real-time PCR kits from PerkinElmer. As a reference point, labs looking to process 1,000 samples in a workday will be looking to use the setup you see on the screen with 1 Chemagic 360 unit, 2 JANUS workstations, and 4 thermal cyclers. The Chemagic Viral DNA RNA 300 kit H96 is designed for automated isolation from 300 microliters of oropharyngeal swab, nasopharyngeal swab, bronchoalveolar lavage, sputum, plasma, or serum using the Chemagic 360 platform equipped with a Chemagic 96 rod head set. The isolation process takes just 60 minutes for 96 samples, followed which the eluted SARS-CoV-2 RNA is ready for subsequent analysis via RT-PCR. The figure here shows an amplification plot from an RT-PCR developed by an end user targeting the RSRP or E gene. The full solution for viral isolation comprises the chemagic 360 instrument, the chemagic 96 broadhead set, and chemagic viral DNA RNA 300 kit H96. As mentioned before, the 96-well format is well suited for high throughput nucleic acid isolation, offers reliable protocols for multiple applications, as well as convenient sample loading and online buffer dispensing. The chemagic 360 uses transiently magnetized rods to attract the beads after binding to the nucleic acids. This feature and the concerted rotation of the rods allow optimal sample mixing during the homogenization and washing steps, thereby increasing the yield and purity of the extracted nucleic acid molecules. The transfer of the magnetic beads between tubes minimizes the possibility of sample contamination between purification steps. The liquid handling steps are met by two configurations of the JANUS instruments are quite straightforward. The first configuration is an automated alternative for a laborious sample reformatting process. The instrument increases sample processing efficiency by reformatting 192 samples in 48 minutes. It also reduces exposure from hazardous sample handling and improves sample integrity by reducing manual errors. The second configuration of the JANUS instrument aids in increasing efficiency by preparing 2 PCR plates in 16 minutes. The instrument reduces hands-on time and, like its sample reformatting counterpart, improves sample integrity by reducing manual errors. On the 24th of March this year, the US FDA authorized emergency use of the PerkinElmer new coronavirus nucleic acid detection kit for detection and/or diagnosis of the virus that causes COVID-19. The FDA also granted PerkinElmer's request to utilize the chemagic viral DNA RNA 300 kit H96 on the chemagic 360 equipped with a chemagic rodhead set 96. We encourage you to visit the FDA's website listed here to access the instructions for use, fact sheet for healthcare providers, and patients for additional details. The PerkinElmer new coronavirus nucleic acid detection kit uses TaqMan-based real-time PCR technique to conduct in vitro reverse transcription of SARS-CoV-2 RNA, DNA amplification, and fluorescence detection. The assay targets specific genomic regions of SARS-CoV-2, namely nucleocapsid N gene and ORF1ab, and leverages multiplexing capacity to enable processing of 94 samples in one run, alongside a positive control and a negative control. The assay includes an RNA internal control that leverages bacteriophage MS2 to monitor the process from nucleic acid extraction all the way to fluorescence detection. The internal control probe is labeled with big fluorescent dye to differentiate its fluorescent signal from SARS-CoV-2 targets. The TaqMan probes for the two amplicons are labeled with the FAM and ROX fluorescence dyes, respectively, to generate target-specific signal. The assay has been optimized to produce results with high analytical sensitivity and specificity. The results confirm a limit of detection of 7.142 copies per mil for the ORF1ab target and 23.380 copies per mil for the N target in oropharyngeal swab matrix using the chemagic 360 platform. The results also confirm a limit of detection of 8.323 copies per mil for the ORF1ab target and 27.246 copies per mil for the N target in nasopharyngeal swab matrix using the chemagic 360 platform. In addition to the significant increase in output from existing factories and production lines, PerkinElmer has set up new manufacturing lines across the globe to meet the growing demand for nucleic acid testing. The latest addition to this capacity expansion plan is the SARS-CoV-2 real-time RT-PCR assay manufactured in PerkinElmer, Finland. This is a CE-marked real-time PCR technique to conduct in vitro transcription of SARS-CoV-2 RNA DNA amplification, and fluorescence detection. Each kit has 4 components with material to process 96 samples in approximately 60 minutes using 14 microliters of extracted nucleic acid as input. The assay includes probes for human RNA target that is used as an internal control to monitor the process from nucleic acid extraction to fluorescence detection. The assay includes probes that, as previously mentioned, cover both the internal control and the targets of the assay. This test is marketed as an in vitro diagnostic device by meeting the requirements of the European In Vitro Diagnostic Directive and is available to ship immediately. In summary, PerkinElmer is very proud and honored for the opportunity to support the laboratory community in this critical time with standardized workflow solutions for high throughput detection of the SARS-CoV-2 virus. To recap, PerkinElmer SARS-CoV-2 testing workflow offers a highly sensitive assay and a comprehensive workflow including chemagic nucleic acid extraction system that can process 96 samples in an hour, and JANUS liquid handling workflow stations that mitigate the risks associated with sample reformatting, isolation setup, and PCR setup, ultimately increasing sample processing capacity. These products can ship immediately, and future availability is backed by a strong supply chain from redundant factories set up across multiple continents. We understand that many labs have adopted a 24/7 operating model. To do our part, PerkinElmer has a dedicated support line that is monitored round the clock to help you in whatever small way we can. Thank you very much for your attention.